don't stop. Don't stop. Don't stop. Don't stop. Don't stop.
Acid-base balance. Introduction. Acid-base balance is very important for the homeostasis of the body, and almost all the physiological activities depend upon the acid-base status of the body. Acids are constantly produced in the body. However, the acid production is balanced by the production of bases so that the acid-base status of the body is maintained. An acid is the proton donor, the substance that liberates hydrogen ion. A base is the proton acceptor, the substance that accepts hydrogen ion. In spite of continuous production of acids in the body, the concentration of free hydrogen ion is kept almost constant at a pH of 7.4 with slight variations. Hydrogen ion and pH. Hydrogen ion, H+, contains only a single proton, positively charged particle, which is not orbited by any electron. Therefore, it is the smallest ionic particle. However, it is highly reactive. Because of this, the H end shows severe effects on the physiological activities of the body even at low concentrations. The normal H end concentration in the extracellular fluid, ECF, is 38 to 42 nm/L. The pH is another term for H end concentration that is generally used nowadays instead of hydrogen ion concentration. The pH scale was introduced in order to simplify the mathematical handling of large numbers. Negative logarithm of H end concentration is taken for calculating the pH as given below. pH equals pK plus log HCO3/CO2. An increase in H plus ion concentration decreases the pH, acidosis, and a reduction in H plus concentration increases the pH, alkalosis. An increase in pH by 1-fold requires a 10-fold decrease in H end concentration, Table 5.1. In a healthy person, the pH of the ECF is 7.40 and it varies between 7.38 and 7.42. The maintenance of acid-base status is very important for homeostasis, because even a slight change in pH below 7.38 or above 7.42 will cause serious threats to many physiological functions. Determination of acid-base status. It is difficult to determine the acid-base status in the ECF by direct methods. 3-2 So, an indirect method is followed by using Henderson-Hasselbalch equation. In this, to determine the pH of a fluid, the concentration of bicarbonate ions, HCO, and the co-dissolved in the fluid are measured. The pH is calculated as follows: pH equals pK plus log HCO3/CO2, where pK is constant with pH of 6.1. Thus, pH equals 6.1 plus log HCO3/CO2. In addition to this, the pH of plasma is also determined by using an instrument called pH meter. Normal acid-base ratio is 1:20, i.e. the ratio of one part. If this ratio is altered, the pH also is altered, leading to either acidosis or alkalosis. Thus, the pH of arterial blood is an indirect measurement of H end concentration, and it reflects the balance of CO2 and HCO3. 3. Regulation of acid-base balance. Body is under constant threat of acidosis because of the production of large amount of acids. Generally, two types of acids are produced in the body: volatile acids, non-volatile acids. Volatile acids. Volatile acids are derived from CO2. Large quantity of CO2 is produced during the metabolism of carbohydrates and lipids. This CO2 is not a threat because it is almost totally removed through expired air by lungs. Non-volatile acids. Volatile acids are derived from CO2. Large quantity of acids are real threat to the acid-base status of the body. For example, sulfuric acid is produced during the metabolism of sulfur-containing amino acids such as cysteine and methionine. Hydrochloric acid is produced during the metabolism of lysine, arginine, and histidine. Fortunately, body is provided with the best regulatory mechanisms to prevent the hazards of acid production. Mechanism. Whenever there is a change in pH beyond the normal range, some compensatory changes occur in the body to bring the pH back to normal level. The body has three different mechanisms to regulate acid-base status: 1. Acid-base buffer system, which binds free H. 2. Respiratory mechanism, which eliminates CO2. 3. Renal mechanism, which excretes H and conserves the bases, HCO. 3. Among the three mechanisms, the acid-base buffer system is the fastest one, and it readjusts the pH within seconds. The respiratory mechanism does it in minutes. Whereas, the renal mechanism is slower, and it takes few hours to few days to bring the pH back to normal. However, the renal mechanism is the most powerful mechanism than the other two in maintaining the acid-base balance of the body fluids. Regulation of acid-base balance. Definition: acid-base buffer system is the combination of weak acid, protonated substance, and a base, the salt, unprotonated substance. Buffer system is the one which acts immediately to prevent the changes in pH. Buffer system maintains pH by binding with free H+. Types of buffer systems. Body fluids have three types of buffer systems, which act under different conditions: bicarbonate buffer system, phosphate buffer system, protein buffer system. Bicarbonate buffer system. Bicarbonate buffer system is present in ECF, plasma. It consists of the protonated substance, carbonic acid, H2CO3, which is a weak acid, and the unprotonated substance, HCO, which is a weak base. HCO is in the form of salt, i.e. sodium bicarbonate, NaHCO3. Mechanism of action of bicarbonate buffer system. Bicarbonate buffer system prevents the fall of pH in a fluid to which a strong acid like hydrochloric acid, HCl, is added. Normally, when HCl is mixed with a fluid, pH of that fluid decreases quickly because the strong HCl dissociates into H and Cl. This is because the H and dissociated from HCl combines with HCO, of NaHCO, and forms a weak HCO. This H2CO3 in turn dissociates into CO2 and H2O. HCl and NaHCO, H2CO3 downward arrow CO2 plus H2O. Bicarbonate buffer system also prevents the increase in pH in a fluid to which a strong base like sodium hydroxide, NaOH, is added. Normally, when a base, NaOH, is added to a fluid, pH increases. It is prevented by adding H2CO3, which dissociates into H and HCO. The hydroxyl group, O, of NaOH combines with H and forms HO. And Na and combines with HCO, and forms NaHCO. NaHCO is a weak base, and it prevents the increase in pH by the strong NaOH. 32333. As sodium bicarbonate is a very weak base, its association with H and is poor. So the rise in pH of the fluid is very mild. Importance of bicarbonate buffer system. Bicarbonate buffer system is not powerful like the other buffer systems because of the large difference between the pH of ECF, 7.4, and the pK of bicarbonate buffer system, 6.1. But this buffer system plays an important role in maintaining the pH of body fluids than the other buffer systems. It is because the concentration of two components, HCO, and CO, of this buffer system is equalated separately by two different mechanisms. Concentration of HCO is regulated by kidney, and the concentration of CO2 is regulated by the respiratory system. These two regulatory mechanisms operate constantly and simultaneously, making this system more effective. 3. Phosphate buffer system. This system consists of a weak acid, the dihydrogen phosphate, H2PO4, protonated substance, in the form of sodium dihydrogen phosphate, Na2PO4, and the base, hydrogen phosphate, HPO4, unprotonated substance, in the form of disodium hydrogen phosphate, Na2HPO4. Phosphate buffer system is useful in the intracellular fluid, ICF, in red blood cells or other cells, as the concentration of phosphate is more in ICF than in ECF. Mechanism of phosphate buffer system. When a strong acid like hydrochloric acid is mixed with a fluid containing phosphate buffer, sodium dihydrogen is formed. Weak acid, this permit only a mild change in the pH of the fluid. HCl and Na2HPO2-NaHPO and NaCl. Strong acid. Weak acid. If a strong base such as sodium hydroxide, NaOH, is added to the fluid containing phosphate buffer, a weak base called disodium hydrogen phosphate, Na2HPO4, is formed. This prevent the change in pH. NaOH and Na2HPO4-H2O and NaHPO. Strong base. Weak base. Importance of phosphate buffer system. Phosphate buffer system is more powerful than bicarbonate buffer system as it has a pK of 6.8, which is close to the pH of the body fluids, i.e. 7.4. In addition to ICF, phosphate buffer is useful in tubular fluids of kidneys also. It is because more phosphate ions are found in tubular fluid. In the red blood cells, the potassium ion concentration is higher than the sodium ion concentration. So, the elements of phosphate buffer inside the red blood cells are in the form of potassium dihydrogen phosphate, KHPO, and dipotassium hydrogen phosphate K2HPO4. Protein buffer system. Protein buffer systems are present in the blood, both in the plasma and erythrocytes. Protein buffer system in plasma. Elements of proteins, which form the weak acids in the plasma are, 1 C-terminal carboxyl group, 2 N-terminal amino group and side chain carboxyl group of glutamic acid, side chain amino group of lysine, imidazole group of histidine. Protein buffer systems in plasma are more powerful because of their high concentration in plasma and because of their pK being very close to 7.4. Protein buffer system in erythrocytes. Hemoglobin. Hemoglobin is the most effective protein buffer and the major buffer in blood. Due to its high concentration than the plasma proteins, hemoglobin has about 6 times more buffering capacity than the plasma proteins. The deoxygenated hemoglobin is a more powerful buffer than oxygenated hemoglobin because of the higher pK. When a hemoglobin molecule becomes deoxygenated in the capillaries, it easily binds with H+, which are released when CO2 enters the capillaries. Thus, hemoglobin prevents fall in pH when more and more CO2 enters the capillaries. Regulation of acid-base balance by respiratory mechanism. Lungs play an important role in the maintenance of acid-base balance by removing CO2 which is produced during various metabolic activities in the body. This CO2 combines with water to form carbonic acid. Since carbonic acid is unstable, it splits into H and NHCO. Entire reaction is reversed in lungs when CO diffuses from blood into the alveoli of lungs. 3H++ HCO, rightward arrow H CO, rightward arrow CO and HO. 32322 and CO2 is blown off by ventilation, chapter 125. When metabolic activities increase, more amount of CO2 is produced in the tissues and the concentration of H and increases as seen above. Increased H and concentration increases the pulmonary ventilation, hyperventilation, by acting through the chemoreceptors, chapter 126. Due to hyperventilation, the excess of CO2 is removed from the body. Regulation of acid-base balance by renal mechanism. Kidney maintains the acid-base balance of the body by the secretion of H and by the retention of HCO, Fig 5.1. Details are given in Chapter 54. 3. Applied physiology, disturbances of acid-base status. Acidosis. Acidosis is the reduction in pH, increase in H and concentration, below normal range. Increase in partial pressure of CO2. Particularly in arterial blood in the body fluids. Decrease in HCO, concentration. Alkalosis. 3. Alkalosis is the increase in pH, decrease in H and concentration, above the normal range, Table 5.2. Alkalosis is produced by decrease in partial pressure of CO2 in the arterial blood. Blood is controlled by lungs, the acid-base disturbances. produced by the change in arterial PCO are called the respiratory disturbances. 2. On the other hand, the disturbances in acid-base status produced by the change in HCO concentration are generally called the metabolic disturbances. Thus the acid-base disturbances are: 3. Respiratory acidosis, respiratory alkalosis, metabolic acidosis, metabolic alkalosis, respiratory acidosis. Respiratory acidosis is the acidosis that is caused by alveolar hypoventilation. During hypoventilation, the lungs fail to expel CO2, which is produced in the tissues. CO2 is the major end product of oxidation of carbohydrates, proteins, and fats. CO2 accumulates in blood where it reacts with water to form carbonic acid, which is called respiratory acid. Carbonic acid dissociates into H and NHCO. The increase H concentration in blood to lead decrease in pH and acidosis. Normal partial pressure of CO2 in arterial blood is about 40 mm Hg. When it increases above 60 mm Hg, acidosis occurs. Causes of excess CO2 in the body. Hypoventilation, decreased ventilation, is the primary cause for excess CO2 in the body. Some of the conditions when increase in PCO2 and respiratory acidosis occur due to hypoventilation are listed in Table 5.3. Respiratory alkalosis. Respiratory alkalosis is the alkalosis that is caused by alveolar hyperventilation. Hyperventilation causes excess loss of CO2 from the body. Loss of CO2 leads to decreased formation of carbonic acid and decreased release of H+. Decreased H and concentration increases the pH, leading to respiratory alkalosis. When the partial pressure of CO2 in arterial blood decreases below 20 mm Hg, alkalosis occurs. Causes of decrease in CO2 in the body. Hyperventilation is primary cause for loss of excess CO2. During hyperventilation, a lot of CO2 is expired through respiratory tract, leading to decreased of CO2. Table 5.4. Metabolic acidosis. Metabolic acidosis is the acid-base imbalance characterized by excess accumulation of organic acids in the body, which is caused by abnormal metabolic processes. Organic acids such as lactic acid, keto acids and uric acid are formed by normal metabolism. The quantity of these acids increases due to abnormality in the metabolism. Causes of metabolic acidosis. Lactic acid. The amount of lactic acid increases during anaerobic glycolysis in some abnormal conditions such as circulatory shock. Keto acids. The amount of keto acids increases because of insulin deficiency, as in the case of diabetes mellitus. In diabetes mellitus, glucose is not utilized due to lack of insulin. So, lipids are utilized for liberation of energy, resulting in production of excess acetoacetic acid and beta-hydroxybutyric acid. Uric acid. The amount of uric acid increases in the body due to the failure of excretion. Normally uric acid is excreted by kidneys. But in renal diseases, the kidneys fail to excrete the uric acid. Some of the conditions when the metabolic acids increase in the body resulting in metabolic acidosis are listed in Table 5.3. Metabolic alkalosis. Metabolic alkalosis is the acid-base imbalance caused by loss of excess H and resulting in increased HCO concentration. Some of the endocrine disorders, renal tubular disorders, etc. cause metabolic disorders leading to loss of H+. It increases HCO and pH in the body, leading to metabolic alkalosis. 3. Some of the conditions when excess H and is lost and HCO content increases leading to metabolic alkalosis are given in Table 5.4. 3. Clinical evaluation of disturbances in acid-base status, anion gap. Anion gap is an important measure in the clinical evaluation of disturbances in acid-base status. Only few cations and anions are measured during routine clinical investigations. Commonly measured cation is sodium and the unmeasured cations are potassium, calcium, and magnesium. Usually measured anions are chloride and bicarbonate. The unmeasured anions are phosphate, sulfate, proteins in anionic form such as albumin and other organic anions like lactate. Difference between concentrations of unmeasured anions and unmeasured cations is called anion gap. It is calculated as anion gap equals Na plus HCO, Cl. 3 equals 144 to 24, 108 milliequivalent/L. Equals 12 milliequivalent/L. Normal value of anion gap is 9 to 15 milliequivalent/L. It increases when concentration of unmeasured anion increases and decreases when concentration of unmeasured cations decreases. Anion gap is a useful measure in the differential diagnosis of the different causes of acid-base disorders, particularly the metabolic acidosis. Variations of anion gap are given in Table 5.5.